So at this point, we know that DNA codes for RNA and RNA codes for proteins, but how does that work? Well, it depends upon a gene. Now, what the actual definition of a gene is has changed a lot over time. The original definition was that one gene was essentially one enzyme, but not all genes code for proteins that become enzymes. So then we said, okay, well, one gene equals one protein, but sometimes it could produce several protein chains or polypeptides. Okay, so one gene codes for polypeptide. Well, sometimes it codes for just RNA. Okay, one gene codes for one product. Many of them produce lots of products. So determining a gene has been a bit of a difficulty over time, but essentially a gene is a specific location on a chromosome consisting of a segment of DNA that codes for a particular protein or RNA molecule that has a function in an organism. Genes are the units that determine how organisms inherit characteristics from their parents. So the central dogma of biology as proposed by Watson and Crick was that DNA can code for itself as replication, that DNA can code for RNA as transcription, and that RNA can code for proteins as translation. And as we know, proteins then code for traits in the organism. All of this depends upon the chromosome. And the chromosome is a structure in the nucleus consisting of long strands of DNA coiled around particular proteins. Each pair of chromosomes has genes that code for the same proteins. That's the point of the homologous chromosomes. And DNA provides the blueprint for the synthesis of proteins. So when a particular protein is needed, the cell must make the protein in the cytoplasm. But there's a bit of a problem there because the DNA can only be found in the nucleus. To send the code from DNA to the cytoplasm's ribosomes where proteins can be produced, you must undergo transcription. Transcription is a process by which a portion of the molecule's DNA is copied into a complementary strand of RNA. Basically, through a series of chemical signals, the gene for a specific protein is turned on, and the code is made from that section of DNA. Unlike replication, transcription doesn't have very many enzymes to keep track of. The most important one is RNA polymerase. RNA polymerase does the job of helicase, it does the job of DNA polymerase, it does the job of topoisomerase, it does the job of single-stranded binding proteins. Basically, the RNA polymerase binds to a promoter sequence further up away from the targeted gene or the transcription unit at a promoter region. In eukaryotes, that binding requires the help of other proteins called transcription factors, and the most common promoter sequence is what's called the TATA box. It's a sequence of TA that is able to allow binding by this polymerase. Prokaryotes don't need any of that because they have a simpler DNA chain, but we do. So that RNA polymerase is used to produce RNA, and I can use different types of RNA polymerase to produce different types of RNA. We know that DNA is double-stranded and RNA is single-stranded, so RNA is only going to be copying one side of this DNA chain. So we call the side that it is copying the template strand. The side that it is not copying is going to have the same sequence as the RNA that is produced because the RNA is forming in a complementary way. The only major difference is that everywhere a thymine would have been, you will now see a uracil. So the coding strand, or the strand that's not being copied, will be exactly the same as the mRNA, except T will be U. RNA polymerase does this in small sections at a time to prevent any major problems and produces what's called a transcription bubble, which is a lot like the replication bubble from DNA replication. It's going to read DNA 3 to 5, but it's only going to build 5 to 3 because, like any polymerase, it's limited that way. But there are absolutely no methods of proofreading. There's basically one error in every 10 to the 5th bases, but it's not really a big deal because you have lots of copies and they don't last very long, so your body doesn't waste the energy to produce perfect transcripts. That overall process of transcription with the RNA polymerase is going to go in these small sections again and again and again until the RNA polymerase reaches a termination sequence. It's not like the promoter sequence, so I can say, okay, the most common is the TATA box. For transcription termination, the method and the way that it's done is very variable. It could be a particular section of code. It could be a loop in the DNA, it could be some sort of functional group. It's of a wide variety of things. But once it has reached the termination sequence, the RNA transcript will separate from the DNA, the RNA polymerase will leave, and the DNA will reattach together. In eukaryotes, there's an additional step. That mRNA is full of junk DNA, and it's not very protected for the rough environment inside a cell. So we're going to have 3 steps for modification that comes after transcription, but before the transcript leaves the nucleus. The first one is the 5' GTP cap will be added to the 5' end. And that is going to protect the mRNA from RNase, which are enzymes that break down RNA, and provide binding energy for it to attach to the ribosome. The poly-A tail on the other end is going to be a long sequence of adenine molecules to provide protection and basically be sacrificial DNA if your mRNA is attacked by an RNase. Introns are non-coding regions of DNA, junk DNA, that do very little for the protein. It turns out they do a lot of things for you, but we won't really go there yet. Exons are the coding regions. So there's these little molecules that mark what's intron and what's exon, and you splice together the exons and remove the introns and basically throw that segment away. That means that the mRNA, the final mRNA, will be shorter than the original pre-mRNA and have no non-coding sequences. Now, I can change what is coding and what is not coding, and that allows for alternative splicing. When you look at humans, we have very little genes compared to many organisms. But because we are capable of alternative splicing, we are actually able to produce a great deal of proteins. Therefore, our proteome, or the sequence of our proteins, is vastly greater than our genome, or the sequence of our genes.